In this video, we're going to talk a little bit about renal functional anatomy. So we want to sort of give you a view of how the structures in the kidney are laid out. And in the kidney, maybe more so than in a lot of other tissues and organs, there's a very, very specific layout to the microstructures, the little things, to make sure that the kidneys are capable of doing their job. And that arrangement is very important to the overall function. So if we start with sort of macro anatomy here, right, we have our kidneys here, sort of in our retroperitoneal spot there in the behind the abdominal cavity, if you will, right? And of course, the urine would leave the kidneys through ureters, right, and then enter the bladder down here. This is the bladder. And then, of course, the urethra is the exit from the bladder. And, of course, we have two kidneys. And so we've got the same thing happening on both sides. Can you function with one kidney? Absolutely. The other kidney, some people are actually born with one kidney, don't even know it. The other kidney will simply essentially double its function to maintain normal excretion. So really there's a bit of redundancy, a bit of backup capacity in renal function, which is good because as we age, we do lose the nephrons of the kidney, deteriorate, we lose nephron numbers, and so renal function does diminish with age. So having two is very helpful, especially in as we age, in old age, right? And so it's not shown here other than our blood flow in and blood flow out vessels here, right? So we'd have, I don't think I have them labeled, but it doesn't matter. We've got a blood flow in and a blood flow out, right? And our blood flow in, that vessel is going to branch and branch and branch until it reaches the vessels that are going to supply the very beginning of the nephron. So the nephron is shown in this dark line here. I'll add some color to it maybe, right? And so we have to have an interface, about 1 million interfaces between the vasculature, the capillaries of the nephron, and individual nephrons. And so the nephron is what's shown here, which I'm trying to highlight a little bit in green. It's not showing up very well, but this guy, right, is the nephron. And it sits partially in the cortex, right? So the cortex of the kidney is this outer layer, right? That's the cortex. It looks like it's just empty here except for this one region. But remember that it would be just packed full of these nephron segments and the glomeruli surrounded by the Bowman's capsule, which is the very beginning of the nephron, in that space, right? So this is chock full of nephron structures and associated blood vessels. And then, of course, you can see that there's a portion that goes down into these pyramid-shaped guys called the pyramids, right? So each of these structures is called a pyramid, right? That's a pyramid. And then in between are columns. So it goes pyramid, column, pyramid, column, pyramid, column, right? And in the columns, it's like where larger blood vessels run and things like that. In the pyramids is where we have, again, more nephron segments, different nephron segments, as well as the associated blood vessels that go along with them. Now you'll notice that, right, when we talked about over here, our ureter sort of leaves the kidney, right? Here's the urine leaving the kidney into the ureter. And so we have to have this hollow space in here, it's actually called the pelvis, into which each one of the pyramids sort of drains. And again, each pyramid would receive the input of thousands and thousands of nephrons, sort of dumping their finalized product, urine, into these open spaces called calyces. And then, of course, the calyces come together to form the pelvis, all open spaces, fluid-filled, urine-filled spaces. And then the urine would drain out through the ureters to the bladder for storage. Okay, so what we want to do now is blow up this picture so we can see sort of the interface between the blood flow and the nephron, and maybe a little bit more detail about how it sits in this space. So let's clear this. right? And what we have here is a blow up. So this guy in dark here is what I had originally in green, if you recall from the previous one. So this nephron starts here. This is Bowman's capsule wrapped around the glomerulus. And then we would have a region called the proximal convoluted tubule up to about there. And then it would lead into the descending limb of Henle's loop. And then we would enter the ascending limb, right, coming back up. Right, this is the direction the fluid's flowing. The sort of the tubular fluid, you can think of it as pre-urine if you like. And that's going to come back up. Notice it went down. Oh, let's do this. Right, this arcuate artery right here, there it is, right, is a demarcation really between cortex from there up and the medulla, which again, you'd be in the pyramid of the medulla from here down, right? And so we have what's called, in this scenario, a long-looped nephron or a juxtamedullary nephron. Again, we'll come back to that later. But these nephrons with the very long loops that go way down into the medulla, some nephrons don't do that. Actually, most nephrons don't do that. But these are really important to overall kidney function because they help, they serve a purpose of helping us conserve water, dramatically conserve water. And again, more detail on that in another video. Once we get back up into the cortex, our ascending loop of Henle, right, we had coming back up here, is going to turn into what's called the distal convoluted tubule, which is reasonably short compared to the proximal. And then it's going to enter this common structure called the collecting duct. You'll notice other nephrons are sort of dumping in there as well, and I have those snipped off, right? But we have other input from other nephrons coming in here or coming in here, right? So they're shared. The collecting ducts are shared amongst numerous nephrons. Probably about 10 to 15 nephrons will join together into one collecting duct. But we generally call the collecting duct part of the nephron, even though there's not one collecting duct per nephron. They're shared. Okay. So we've got our nephron structures here. We can kind of see how it fits in. Now let's follow the blood flow for just a second, though, right? Because remember, we had talked about how that blood flow comes into, right? So our larger branches of arteries are coming into the kidney. And here they go, and they're going to send blood out into the cortex. And that blood out of the cortex is going to go off and branch into these smaller and smaller vessels, right? And eventually we reach what's called the afferent arterioles, which lead directly into this glomerular capillary bed right here. This little tuft, this ball of capillary is called the glomerulus. And again, you can see the label here. Hopefully you can read that. Bowman's capsule surrounds the glomerulus, and the whole structure is called the renal corpuscle. And then if you'll recall, blood flows back out of the glomerulus and then breaks up into a second capillary bed shown here. Right, so this vessels are crisscrossing and such. Right, and then into what will eventually become, and I should maybe add or make this sort of become bluish, because remember it's going to become venous blood and we will have given up oxygen along the way. Right, so let's make it a little bit bluish. Okay, and again, the peritubular capillaries exist to absorb everything being reabsorbed from the nephron, right? So what's coming out of the nephron, and the peritubular capillaries are kind of everywhere. I'm just showing them in one spot here. But what's reabsorbed goes into them, and remember what's secreted would come out of them and go into the nephron at a variety of locations. Okay, now let's take an even closer look and label the individual nephron segments again, and then especially at the glomerulus, so we can see some key structures that will be very intimately tied to particular kidney functions. Right, so for this one, I'm going to draw our incoming vessel, the afferent arteriole. I'm going to draw just sort of loops here to indicate the glomerular capillaries. Then there's the vessel that exits the glomerulus. That was the efferent arteriole. Then we have our peritubular capillaries. And have these sort of branching. Right, so those are the peritubular capillaries, PTCs. Right, we had the, I'll do some abbreviation. Well, I'll write this one out. So this is the afferent arteriole. Remember, a million of these, right? So the afferent arteriole is our inflow vessel into the glomerulus. Think A for excess. The cluster of capillaries here was the glomerulus. Remember, that's singular. Glomeruli is plural. And then the glomerular capillaries come together to form, right, so this is afferent arteriole. The glomerular capillaries come together, coalesce into the efferent arteriole. That's this guy. Think E for exit, right, coming out of, so this is flow is going this way. Right, and that's really our portal vessel between the two capillary beds, the glomerulus and the peritubular capillaries. Okay, now our nephron, and I'm going to shrink this a bit because it's awfully long and I want to run out of room. But our nephron starts by wrapping around the glomerulus, and then it wiggles, right? Convoluted means sort of wiggly and bendy, right? And then it's going to go into, and I'm drawing this in a way, well, in a lot of ways, but in one key way, not anatomically correct, and I'm going to point out in a minute. Right? Then we go down and we form a loop of Henle. The way down is kind of thin, the way up is kind of thick, largely. And then there's another shorter wiggly part at the end, and then we would end up going into the collecting duct. Remember, collecting ducts are shared, so we'll put another one coming in here. And so at the bottom here, this ends, and that's where urine would exit the collecting duct. Going this way. Right, so let's label our segments here, right? So this was Bowman's capsule. Right, this guy, this first segment of nephron, is the proximal convoluted tubule. You should be very familiar from lab if you have lab right now. Then we're into the, right, we're going this way. So that's descending, then we're going back up, so that's ascending, so that's the descending and ascending limbs of the loop of Henle. Right. And then, as I mentioned a minute ago, we enter the DCT, distal convoluted tubule. Remember what proximal and distal mean. Proximal means closer to the point of origin or the beginning of something. Distal means farther away. Right, so the PCT is proximal because it's close to the glomerulus, renal corpuscle, the whole thing together. Distal is farther away. And then, of course, the last segment here is the collecting duct. All right, now what's not, oh, and one other thing I should point out is that right about here was, remember, the loop, and again, for some nephrons that have long loops. The collecting duct always goes down into the medulla. Remember, so below is the medulla. The glomeruli, Bowman's capsule, the PCT, and the DCT are always in the cortex. Right? Longer loops are in the medulla if we have a long loop like this, but the collecting ducts are always in the medulla. All right. Now, we have to come back to the beginning here because we need a little bit more detail. And And what we're missing is part of a regulatory system that helps us adjust. Remember, one of the key things that's happening here is dramatic filtration. Right? That is the function of the glomeruli, is to generate a filtrate due to the Starling forces driven by the primary filtrative force, which is PC. Let me label that here. So PC is the capillary hydrostatic pressure. That's the guy that's tending to win and therefore drive filtration into the nephron. Now, what's the bit that's really anatomically correct here is that right at the end of the ascending limb of Henle, or some people say the beginning of the DCT, it's kind of like right at that spot, that is not actually over here by itself. This thing actually is flopped over such that this spot, I just draw the line through right here, actually sits right here. Right? So this is the tube coming in and out of the screen. So there's a hole in the middle of this tube, right? There's the inside of the tube. Now we're going to draw the cells, right? They're cuboidal epithelial cells, so they're kind of block-like here. I'll put some nuclei in them so we can tell that they're cells, right? And then this area, this hole in the middle is the lumen, the tube, the inside of the tube where the tubular fluid is flowing. Again, it's almost urine, but not quite. Right? And so that is part of the, again, beginning of the DCT or end of the ascending loop of Henle. I like to say end of the ascending loop of Henle. I think that's more accurate. But that has a name, which is the macula densa. And again, without getting into too many details, the macula densa senses what's happening sort of at the end of the loop of Henle and can tell the beginning of the system whether it needs to filter more or filter less, right? And so it can read, basically saying, read certain parameters, the flow rate and the contents of the tubular fluid to say, oh, we need to filter more or we need to filter less. And we filter more or filter less by adjusting, you guessed it, PC. PC is the Starling force that we can regulate to adjust GFR, glomerular filtration rate. That's this F, right? This F equals GFR. Filtration is just the generic term for any capillary, but when it's at the kidneys and it's the glomerular capillaries, we call it GFR. Right? And so what we need to do to adjust PC is we adjust the resistance of the afferent relative to the efferent arterioles. Okay? And so the macula densa will help us do that, and the macula densa will actually adjust the contraction of the smooth muscle. Again, more detail later in a separate video. The smooth muscle surrounding this afferent arteriole. Now, there's smooth muscle surrounding the efferent arteriole too, and so we can regulate that as well, and there's a particular hormone that does that very effectively. Right? But so the macula densa is this key regulatory structure. All right, well, it can do that directly through a paracrine signal. In other words, these cells are right next to those cells, the cells of the afferent arteriole, and it can send a chemical signal that will cause them to adjust their contractility. Okay, that's one mechanism. Another mechanism is that these cells of the afferent arteriole, I'm going to colorize a couple of these. Every other, stand out, right? These guys are called juxtaglomerular cells. Often just shortened to JG cells. Well, these are the guys that produce an enzyme called renin. And renin, through a sequence of actions that get stimulated, will result in the production of a hormone called angiotensin 2, shortened or abbreviated ANG2. Okay, and as we said in the previous video, angiotensin 2 is really important to both renal function by stimulating other hormones, but also to cardiovascular function because it's a major, very potent vasoconstrictive hormone, right? So it's going to adjust blood pressure. All right, the last thing we want to point out, just in terms of sort of anatomical designations here, is that the macula densa and the JG cells kind of work together because the macula densa signals can be direct to adjust the contractility of the afferent arteriole, but they can also be indirect and act through the JG cells to stimulate renin and angiotensin 2. And guess where angiotensin 2 is going to act? And again, foreshadowing another video, going to act to adjust, i.e., in this case, constrict the efferent arteriole. And remember, what are we doing if we constrict the efferent arteriole? We're increasing the PC, the hydrostatic pressure, upstream. The macula densa directly actually dilates the afferent arteriole, which guess what that does? Increases PC, right? You open this up and allow the pressure to enter the glomerular capillaries, then the pressure there rises and filtration goes up, right? And that is a lot of controlling what's called single nephron GFR, right? SNGFR. Now, what I was doing over here with this labeling is that this whole thing is called the juxtaglomerular apparatus, or also shorthand JGA. Right, so the JGA is the macula densa plus the JG cells, all working together to either locally or hormonally control GFR. Right, locally through paracrine factors, hormonally through angiotensin 2. Okay, so I wanted to tie in this video. I wanted to tie together not just the anatomy with some of the key functions of that anatomy, right? We don't have it all here, but I wanted to, there's some very specific anatomical, arrangements here with the macula densa being adjacent to the afferent arteriole and these JG cells that's very important.